The IonFlux system is an automated solution for high-throughput ion channel drug discovery and research. Using Flexion's Well Plate Microfluidic technology, the IonFlux delivers high-quality patch clamp data using standard well plate formats. This system offers numerous benefits to labs of any size, including fast compound addition for ligand and voltage-gated channels, a small benchtop format, and precise temperature control. Furthermore, the IonFlux eliminates the technical learning curve associated with the manual patch clamp technique and provides seamless integration to high-throughput screening workflows. Ion channels play a critical role in the regulation of the central nervous system and the physiology of nerves and muscles. As a result, they have been implicated in numerous disease conditions such as diabetes and epilepsy, and are leading targets for new medications. The patch clamp method is the gold standard for evaluating ion channel activity in vitro. When implemented in a manual fashion, the patch clamp assay is limited to around 10 data points per day, which is well below the threshold needed for meaningful drug discovery and development efforts. The IonFlux platform uses the proprietary Well Plate Microfluidic technology to automate the patch clamp method in a convenient and high-throughput well plate format that comes in 2 scalable configurations to accommodate a broad range of applications, including both voltage and ligand-gated ion channels. The IonFlux HT is suited for high-throughput screening in drug discovery and development. This version is capable of producing up to 10,000 data points per day. For additional throughput, up to 4 IonFlux HT systems can be arrayed around a single liquid handling robot for up to 40,000 data points per day. The low cost per data point and minimal compound usage make the IonFlux HT ideal for drug screening, drug development, safety toxicology, and ion channel research. The IonFlux 16 is a cost-effective solution for moderate throughput applications such as cell line development, mutant screening, and assay development. For the first time ever, users have access to true automated patch clamp technology at a comparable cost to a manual patch clamp rig. The IonFlux system is comprised of a benchtop instrument, a proprietary microfluidic device in an SBS standard well plate format, and a complete acquisition and analysis software package. Unlike other automated patch clamp instruments, the IonFlux reader maintains a very small footprint which closely resembles a plate reader. It easily fits on the benchtop and has available integration with commonly used laboratory automation robots, enabling the IonFlux to fit seamlessly into existing high-throughput screening workflows as well as research laboratories. The IonFlux utilizes ensemble recording, which means that a cohort of 20 cells are patched in each recording zone and their currents are averaged to produce a single data point. This approach produces a high success rate and minimizes the effect of cell variability. Each 384 well plate can process 256 unique compounds and produce 512 data points. The 96 well plate used with the IonFlux 16 processes 64 compounds and produces 128 data points. Having the ability to run high-throughput electrophysiology screens opens the door to a wide range of ion channel experiments. Common applications include experiments with ligand-gated channels such as the GABA and nicotinic receptors, voltage-gated channels such as the sodium and potassium channels, and safety toxicology focusing on the HERG potassium channel. Mutant and cell line screens can also be performed. Furthermore, the IonFlux enables analysis of the functional expression of ion channels. The IonFlux system utilizes microfluidic devices called IonFlux plates, which feature the proprietary Well Plate Microfluidic technology. Plates are available in SBS standard 96 well and 384 well formats. Each device utilizes an array of microfluidic channels positioned underneath the wells to conduct high-throughput electrophysiology measurements. To begin an experiment, all the reagents are added to the IonFlux plates. Each device is divided into experimental patterns, with each pattern having specific wells designated for cells, compounds, and recording buffer solutions. The 96 well plate has 8 experimental patterns, while the 384 well has 32 patterns. Reagents can be added by using a liquid handling robot or a manual pipette. Once the plate is formatted with the cells and reagents, it is placed on the tray and loaded into the instrument. Once inside the instrument, a pneumatic interface seals against the top of the plate, which provides regulated air pressure and vacuum into the wells. The interface also contains an array of electrodes which dip into specific wells on the plate involved with the recording. The plate is initially primed with fluid by applying positive pressure to all the channels. Once priming is complete, the cells are introduced into the main flow channel by applying positive pressure to the cell input well. Cells are trapped by applying negative pressure to the wells connected to the trapping zones. The cells get trapped at small junctions off the main flow channel, which are similar in size to manual patch pipettes. The cells attach and seal to the traps much like a conventional patch pipette. Once the cell is attached, whole cell voltage clamp is achieved. The IonFlux system uses ensemble recording to provide maximum success rates. Each trapping zone captures 20 cells, which are used to make one patch clamp recording. There are 2 trapping zones per experimental pattern. Across the entire plate, there are 64 parallel recordings in the IonFlux HT and 16 parallel recordings for the IonFlux 16. Once the cells are trapped, the IonFlux instrument and software can apply voltage sweeps across the electrodes and measure the ionic current through the cell membranes. After recordings are established, up to 8 different compounds or concentrations can be applied to the cells in rapid succession. The fast compound addition makes the system suitable for both ligand and voltage-gated channels. In between compound additions, the compounds are washed away while the system continues to record. Because of the unique nature of the fluidic perfusion system, many workflow options are available, including cumulative responses and compound repeats. Once the workflow is complete, the plate is ejected from the system and ready for the next experiment. The IonFlux system comes with an advanced software package for data acquisition and analysis. Once the software is loaded, the user will see 3 primary screens: Setup, Runtime, and Data Analysis. The Setup mode contains all of the controls needed to define an experiment. In this mode, experiments are initialized and whole or half plate is designated. The table of utilized compounds is also imported in Setup mode. In the Experimental Sequence tab, parameters are set for the 4 phases of the experiment: prime, trap, break, and data acquisition. Here, the voltage protocols are defined. The runtime mode shows the data as it's being collected. The real-time sweep window displays currents generated during each voltage pulse for every channel. Experimental progress is also reported. The data analysis mode enables quick review and analysis of completed experiments. Tools are provided for viewing sweep and trace data, as well as for logging specific events such as compound introduction. A heatmap module helps identify the most promising compounds in the screen. All data can be easily exported in .csv format to Excel or other spreadsheet applications. The data can be directly exported to associated Excel templates for automated data triage, normalization, plotting, and fitting of EC50 values. A wide variety of experiments and target screens can be performed on the IonFlux system. Here, an example is demonstrated to assay for modulators of the human GABA-A receptor expressed in HEK293 cells for millipore. This ligand-gated chloride channel is involved in CNS inhibition, thus constituting an important drug target in epilepsy, anxiety, and insomnia. To perform screening assays, cell ensembles were exposed to EC20 concentrations of the native ligand GABA in the presence of ascending concentrations of modulator substances that bind at the allosteric site on the receptor. The plots show dose-response curves for 2 positive allosteric modulators in the benzodiazepine class, namely triazolam and diazepam, for which EC50 values of 3 nanomolar and 0.5 micromolar were obtained. The modulator-dependent shift in the apparent GABA affinity was then determined by cumulatively applying different GABA concentrations in the presence of a fixed concentration of the modulator, in this case 3 micromolar diazepam. This type of experiment is enabled by the flow-through fluidic design available in the IonFlux instruments. The EC50 for GABA was shifted by about 1 micromolar to lower concentrations due to the increased binding affinity in the presence of the modulator. The IonFlux instrument also facilitates other pharmacological experiments, including the example shown here, typical for blockers of the Neuronal HNAV 1.8 sodium channel, a voltage-gated channel involved in the perception of pain. In this assay, the sodium channel, expressed in HEK293 cells, was activated by voltage clamp steps from -120 mV to 0 mV applied every second. This protocol therefore provides information on tonic block of the channel, since the channel re-enters the rested state between depolarizations. Cumulative blocker dose-response curves are illustrated for the local anesthetics lidocaine and tetracaine, providing IC50 values of 320 micromolar and 34 micromolar respectively. In a second graph, the lidocaine affinity for HNAV 1.8 is compared to that for HNAV 1.7, another voltage-gated sodium channel involved in pain sensation. The IC50 for HNAV 1.7 was found to be 3 millimolar, revealing a 10-fold lower affinity when this channel binds lidocaine, compared to that of HNAV 1.8. The IonFlux instrument can also be used for safety pharmacology applications such as HERG screening. The HERG potassium channel has been implicated in adverse cardiac events associated with diverse drugs on the market, leading to their withdrawal, and it is now required that all new drug compounds be screened against this channel. Blocker dose-response curves were performed for characteristic voltage dependence HERG channels expressed in CHO cells for 3 commonly used reference compounds, namely terfenadine, cisapride, and quinidine. The graphs show the typical current profile obtained when HERG channels are activated by a 2-pulse protocol, which is necessary because of its rapid inactivation rate, as used to reveal HERG tail currents. The graphs show the activation voltage dependence and rectification properties of the HERG current recorded in the IonFlux instrument. These recordings closely replicate accepted channel behavior represented in the literature. The blocker dose-response curves reveal IC50 values for the 3 reference compounds of 24 nanomolar, 60 nanomolar, and 0.64 micromolar. In conclusion, the IonFlux enables a wide range of ion channel screening experiments for pharmaceutical, biotech, and academic laboratories. Key advantages include throughput of up to 10,000 data points per day per reader, up to 40,000 data points per day with the multiplexing option, fast compound addition for ligand and voltage-gated channels, a small benchtop format with plate reader simplicity, and precise temperature control. The IonFlux also eliminates the technical learning curve associated with the manual patch clamp technique and provides seamless integration to high-throughput screening workflows. Please visit www.fluxionbio.com for more information on the IonFlux system or call to speak with a product representative.